Access granted. Access granted.
Transgenesis refers to the process of introducing transgenes, in example, an exogenous genes from one organism into another, with the intent of enabling the latter to exhibit a new properties that can be transmitted to its offspring. It can be done by using liposome, plasmid vectors, viral vectors, pronuclear injection, protoplasm fusion, and ballistic DNA injection. Therapeutic protein is technically any protein-based drug. The term was first used to describe medicine that are genetically engineered versions of naturally occurring human proteins. These proteins are produced by sewing microbial fermentation on cell culture, in transgenic animals, and also in transgenic plants. Recently, the therapeutic proteins categorized in terms of their functions and therapeutic application. Therapeutic proteins was placed in groups of either approved by FDA or investigated in vivo or in vitro. Briefly, the proteins that are used to treat metabolic disorder or endocrine dysfunctions like interleukin-1 receptor antagonist for type 2 diabetes mellitus and disorder like hemophilia A, which require clotting factor A, have been included in Group 1A. Group 1B includes the proteins that are known to stimulate various hematological and body immune response, such as interferon alpha for the treatment of hepatitis C and erythropoietin for the chronic anemia. Proteins that have been therapeutically used to alter the pathophysiology of a disease are included in Group 1C. Some examples are botulinum toxin type A and B for multiple dystonia and lepiridine for heparin-induced thrombocytopenia. The proteins that have been classified under Group 2A are known either to stimulate signaling pathway or inhibit the functioning of molecules or organisms via direct binding. Compounds that have the ability for targeted specific delivery of proteins have been included under Group 2B. In Group 3, it contains proteins that are used for vaccine. Lastly, proteins that are members in Group 4 used for diagnostic purposes such as infectious disease diagnostic and imaging agent for the cancer detection. Vaccine production has several stages. Firstly, inactivation process. This involves making of antigen preparation. Next, in purification process, the isolated antigen is purified. Last step is formulation. In this process, the purified antigens is combined with adjuvants, stabilizers, and preservative to form final vaccine preparation. Therapeutic proteins are used to treat patients suffering from cancer, heart attack, stroke, cystic fibrosis, diabetes, anemia, and hemophilia. Being delicate compounds, most of the commercially available therapeutic proteins are administered mainly by the parenteral route.